In the blood spatter experiment, we're looking to see how height affects blood spatter when it hits the surface. We're doing this experiment to see if our victim, Anna Garcia, was standing and someone hit her, or if she fell and hit her head while on the floor.
Different blood types generate different antigens and antibodies. Type A blood contains A antigens and B antibodies. Type AB blood contains A and B antigens but no antibodies. Type B blood has B antigens and A antibodies. However, type O blood has no antigens but A and B antibodies. Blood antibodies prohibit the existence of that blood type in its system. For example, someone that has A blood type can receive type B blood transfusion, since the person's immune system will attack each blood cell. Type O blood, however, can be transfused into anyone, no matter their blood type. DNA is what contains genetic information exclusively for each organism.
DNA is made up of millions of nucleotides. Nucleotides consist of a phosphate group, a 5-carbon sugar, and a nitrogenous base, which could be either adenine, thymine, cytosine, or guanine. DNA has a double helix shape, meaning there is 2 strands of nucleotides that come together at the middle, forming a shape of a twisted ladder. When the nucleotides come together, adenine only pairs with thymine and cytosine only pairs with guanine.
This is a red blood cell. It has A antigens sticking out of it, but how are those made? Everything starts at the nucleus of the cell, where transcription occurs. Transcription is when DNA unwinds with the help of 2 proteins, polymerase and helicase. These proteins separate each strand of DNA to let one of them be completed by the messenger RNA. RNA, like DNA, is also made up of nucleotides, but instead of thymine, RNA has a nitrogenous base that it replaces it, called uracil. When transcription is complete, mRNA travels outside of the nucleus to a ribosome that is floating in the cytoplasm. That's where translation occurs. Ribosomes separate mRNA by codons, 3-letter sequences. They code for a specific anticodon that transfer RNA brings into the ribosome. tRNA are connected to a specific amino acid that mRNA codons code for. tRNA keeps bringing amino acids until the mRNA tells it to stop. When it stops, a polypeptide chain is formed. They're also called proteins. Lastly, these proteins go through the Golgi apparatus and then outside of the cell, sticking into its surface, creating A antigens. Going back to our experiment, we know that the evidence found in the crime scene, it's blood. Now we have to test our suspect and compare the DNA found in their blood with the blood at the scene.